Okay, so welcome to part two. So again, we talked about enzymes, and the key features is their active site. I know I keep saying this. If I keep saying it, it's because it's important. Um, so it's got the active site right here that matches the substrate, and that's the lock and key model says they match exactly. The induced fit model basically says as the substrate gets closer to the active site's shape shifts to allow the substrate and the enzyme to bind. And again, that shape of that enzyme is ultimately determined by the primary structure of the protein, because they are proteins, which is determined by the sequence of amino acids, which is ultimately determined by the sequence of DNA. Okay? So we talked about enzymes and the role in metabolic pathways, and the fact that each kind of step in a reaction has an enzyme that allows us to control the different stages of your body to control different stages of that pathway or that cycle or that chain. um, and again, the role is basically they increase the rate of reactions, they speed up reactions, they lower the activation energy, so they make the reactions more likely to happen, and the specific enzymes allow us to control those pathways. So again, a couple other things. Enzymes catalyze reactions, so they speed them up. They're substrate specific, and they lower the activation energy. And we talked about the collision theory of enzyme. It's basically all a bunch of random molecules bumping into each other. And we kind of talked about how this works. Don't worry about the immobilized enzymes yet. We talked about how activation energy is lowered in enzymes, making the reactions more likely to happen. We looked at the steps, right? So the substrate collides with the active site in the right shape and orientation. That creates the enzyme substrate complex. The chemical reaction can occur, and we get the products here. But notice the enzyme doesn't change shape. Induced fit model basically works in a very similar way, except that the active site here changes shape to match that. Okay? Then we started. Sorry, we started talking about with pH, temperature, substrate concentration. How can we change how these work? So, first is kind of this statement. Um, Basically, enzymes are somewhat fragile. Remember back when we talked about protein folding and the shape of the protein? It's held together by intermolecular forces, hydrogen bonds, van der Waals interactions, those sorts of things. Um, with that, those bonds aren't chemical bonds. They're fairly weak individually. So what that means is enzymes are affected by pH, temperature, and what can those two can lead to is the protein being what we call denatured. Okay, so a denatured protein looks like this. Okay, and you can see the problem right away in this picture is if you change the shape of the protein, you change the shape of the active site right here, right? That's the active site. And with that active site changed, the substrate no longer fits. So what happens here is you get no reaction. Okay, it doesn't change. It doesn't catalyze that reaction anymore. The enzyme no longer works. It's denatured. It's changed shape. And that can be pretty serious. There's lots of things that can cause that, but the ones we talk about is a change in pH and temperature. Okay, so pH is kind of relevant in your stomach. For example, your enzymes that break down proteins in your stomach work at a very acidic pH. By the time they get to the small intestines, those same enzymes don't work anymore because the pH is wrong. Alright, so one other thing you need to be able to do is draw and explain graphs of temperature, pH, and substrate concentration. So kind of zooming in here, this says enzyme activity. So this is basically the rate of reaction, okay? And this is the change in temperature. So kind of here where you have, sorry, my bad, non-straight line. Right here, whatever volume this temperature is what we call the optimum, temperature. So for example, in the human body, it is approximately 36 to 37 degrees Celsius, okay? So our enzymes in our body are designed to work at that temperature. Now, if you lower that temperature, basically what you're doing is you're taking energy out of the system, it's colder, and these molecules move around less. Now remember, enzymes were all just a bunch of random reactions, right? So if those molecules are moving slower, which is especially true when the temperature is lower, then they're not going to collide and smack into each other as often. Okay, so the idea that the substrate and the idea that the substrate and the enzyme up here match up doesn't happen as much when it's colder. Okay. Now, if we go the other way, and it gets too hot, notice the drop-off's pretty steep, right? Here, it denatures, right? Remember, we have the opposite problem. The enzymes will, the substrates and the enzymes will move around faster when it's hotter, but eventually what happens is the bonds that keep this shape, the intermolecular forces, hydrogen bonding, van der Waals interactions, and so forth, these break down, the shape changes. So over here, kind of when we get over to this point, we've denatured the enzyme, okay? So the enzyme will no longer work. So that's kind of the one explanation for that graph, and it's kind of all explained down here. Now, pH, again, very similar idea. Here's our optimum pH, okay? So, but as you move away from that optimum pH, the acidity or the acidity changes, and what that can do is that can disrupt the bonds holding the shape of that enzyme. So we're right back to this thing again, right? The shape of the enzyme changes so that the active site is no longer valid, and the result is that the enzyme and the substrate don't fit, they don't match. So the enzyme can no longer speed up the reaction. All right, and the very last graph here is enzyme activity and substrate concentration. So this one's should make sense. So after a while, if you increase the substrate concentration, you will increase the rate of reaction. The reaction will go faster. That makes sense because, remember, we're all a bunch of chemical reactions. If I have 3 enzymes and the blue are the substrates, the more substrate I have, the more likely the enzyme and the substrate are going to bump into each other. If I increase that even more, so I add even more, then it's even more likely, right? So here's kind of this spot on the graph. Here's maybe this spot on the graph. But let's say I want to go really wild, and I have 3. And let's say I add lots and lots of substrate, right? I want this reaction to go as fast as I can. I want to make whatever it is very, very quickly. So I add a whole bunch here. The problem is you've added all these, but you haven't changed the enzymes, right? So the way I kind of ask students to think about this, imagine your 3 enzymes and they're undergoing chemical reactions. Imagine if each substrate lined up, waiting in line to be catalyzed, right? The more substrate you add in each of these conditions, all you're ending up with is more lines. Now here and here, it's okay because the enzyme can still work really, really fast. But by the time you get over to this point, it doesn't matter how many more you add. It doesn't matter how fast the enzyme's working. You're still going to have a ton of substrates behind it waiting, right? What you need to do here to lower this line is add more enzyme, but we're not going to talk about that, right? So the idea here, I think about this is like a cashier in a grocery store, right? Especially now with the coronavirus, imagine everybody buying toilet paper. If you only have 3 cashiers open, it doesn't matter how fast they go. The limitation is all the people waiting in line with the toilet paper, for example. Right? The only solution is to open more registers. But even if that's all the registers you have, it's not going to go any faster. Okay? So basically the idea here is all the active sites of the enzymes are full. Okay? So all the active sites are full. So that's kind of temperature, pH, and substrate concentration. There's some explanations down here. I think they're good. So that'll help you too. Now the other two, okay, so like here would be the end of SL stuff. So now over here we're going into the HL stuff. You can also have what we call competitive and non-competitive inhibition. Okay, so with the idea of inhibition is, again, this slows down reactions. Okay, so in competitive inhibition, what you see here is the substrate, the active site, right, lock and key. But what you see over here, this is a chemical reaction. What you see here in the inhibition, especially with competitive, is the inhibitor, which is right here, looks a lot like the substrate, but not quite. So the shape down here is the same, but then it's different. So what happens with a competitive inhibitor is it binds to the active site, but it stays, generally speaking. All right? It doesn't leave. Okay? So basically you've disabled that enzyme. You've blocked the active site and the substrate can't react with it. So this will cause the rate of reaction to decrease because you're removing enzymes available from it. It's blocking the active site, right? And that's why you kind of call it competitive. It's the substrate and the active site. Substrate and the inhibitor are both competing for the active site. Okay, we're going to contrast that with the idea of non-competitive inhibition. So in this case, what happens is we have what we call an allosteric site. Okay, so it's another site on the enzyme that has kind of a particular shape. Okay, so normal everyday kind of reactions here. Substrate binds with the active site, everything's fine, the reaction occurs, we're all good here. But what can happen with a non-competitive inhibition is it actually binds at the allosteric site. But notice when it bound here, the enzyme, the active site changed shape. Okay, so the active site changed its shape. So what happens now is the substrate no longer fits. Okay, it no longer matches up, and that's because of the allosteric site being blocked with the inhibitor. Okay, this is actually very common. This is kind of basically a way that we can turn on and turn off enzymes in a whole field, like in a whole series of chemical reactions, right? Sorry, let me adjust it. So in a whole field of chemical reactions, so A makes B makes C makes D, what often happens here is D inhibits the enzyme responsible here. Okay, so it turns off that enzyme, because if you have too much of D, you don't need C, you don't need B, so it turns it off there. And it usually does it this way. And we'll look at some examples later. But we say that this is non-competitive because they're not competing for the active site. In this case, the inhibitor doesn't have to fight the substrate, so to speak, for the same site because it's got its own site. Right? If these both are competing and you just increase the amount of substrate, then the substrate is going to bind with the active site more often. The inhibitor doesn't care about the active site. Sorry, the non-competitive inhibitor doesn't really care about the active site. It binds with the allosteric site. So it doesn't matter how much you increase the substrate, they're not competing for each other. Okay, so a couple of interesting things here. So if you look at the rate of a reaction with no inhibitor, it kind of tends to look like this. With a competitive inhibitor, if you increase the substrate concentration, you can overcome the effect of the inhibitor. This kind of makes sense. Okay. Now, however, with a non-competitive inhibitor, it doesn't really matter how you increase the non-competitive inhibitor, or how much you increase the substrate, because so the graphs look different. So the non-competitor tends to become a bit more constant a lot quicker than the competitive inhibitor. So then down here, I just talked about some examples. You do need to be familiar with some examples. The first one's Viagra. Right, this is for people who have trouble, males who have trouble getting an erection, and it talks about there's an enzyme called PDE5 which breaks down a molecule that increases vasodilation, that increases blood flow. So the active ingredient in Viagra inhibits that enzyme, which then promotes vasodilation, which then helps lead to and maintain an erection. Far more information you guys wanted to know, but there you go. Penicillin, we talked about, is an antibiotic, and it prevents a chemical reaction that cross-links the cell walls of bacteria, an enzyme called DD transpeptidase, which helps make a bacterial cell wall. So it inhibits that through non-competitive inhibition, and the bacteria can't make a cell wall, and that means they can't survive. Okay? And before you ask, yes, you need to know specifics about each of these examples.